Okay, so this video is going to be about DNA binding proteins. There we go. So starting off, I'm going to be talking about how histones bind to DNA specifically to create kinks and create these little circular bends in the DNA. And so what we have here is just a kind of half a representation of the histone 3 and the histone 4, and these form a dimer whenever you're forming nucleosomes. And so the contacts that we're making here are all going to be electrostatic interactions, and it's going to be between DNA's negatively charged sugar phosphate backbone, which means that it's going to be binding in the minor groove, and it's going to be between the partially positive N-terminus of the histone proteins, since there is a helical dipole in the alpha helix of the secondary structure, and it causes the DNA to kink at very specific sites. And so not only does it bind at the N-terminal region where you have that partial positive in the alpha helix, it will also bind to the positively charged residues in the proteins, such as arginine, which are all labeled here, and also positively charged proteins like this lysine at the very side. And so these are still making these electrostatic interactions between the positive parts of these proteins and the negative charges of the sugar phosphate backbone. And another thing to notice here is what you see with the histones is that they have very similar structures that are usually always conserved, where you have this helix, loop, helix, loop, helix. And that's present for both histone 3 and histone 4, and it can also be seen in the other histones as well. So that allows it, again, to keep this helical dipole. It allows it to kink the DNA at specific sites and create these electrostatic interactions with it, which is where the DNA will bind to the protein. So next we're going to be talking about how it makes sequence-specific DNA binding. Before I was talking about just the electrostatic interactions between the negatively charged part, which would be in the minor groove. So it was completely sequence independent. Although it was at specific sites to kink the DNA, it was only at sites in the minor groove where you have the negative charges. It wasn't interacting with any of the bases within the DNA sequence. So now we're going to be talking about the proteins that will bind to at sequence-specific sites. So it's going to be in the major groove in this case. So the main part that we're going to recognize is going to be this helix turn helix. And this recognition helix is going to be the one that interacts with DNA. And it's also going to undergo hydrogen bonding with the nucleotides, but it could also undergo some electrostatic interactions. In this case, the protein isn't going to be hydrophobic and the DNA isn't hydrophobic, so the hydrophobic effect doesn't really apply in these scenarios. So although the helix turn helix is the main idea, which we also see here whenever we think about sequence-specific DNA binding proteins, you can also have other kinds of alpha helices like these leucine zippers or any of these alpha helical domains, but you could also have anti-parallel beta sheets. And you can see that like the beta barrel down here or this beta sheet binding. Primarily, it's going to be alpha helices, but you can also see beta sheets, and there is a wide variety of types of proteins with different secondary structures that can bind to DNA. The major thing is that if it's sequence specific, it will always bind in the major groove. So next is about how exactly these proteins are going to bind to the target sequences, because you have this extremely long stretch of DNA. Sometimes it's being interacted on by other proteins. It could be bound up in nucleosomes where it's bound to histones. So there's really three major ways that it could bind. And so the first one is represented by this little green protein over here, where it could bind directly to the target sequence. So this is going to undergo 3D diffusion. You have this entire stretch of DNA that's in front of you, and you have to only bind to this target sequence. And so you have to overcome this K off versus the K on and make sure that it's super specific, and also understand how tightly it binds for it to actually create this target sequence and have this interaction. So this isn't extremely likely. You probably have a really low probability of this protein actually consistently binding to this target site efficiently. So this is probably the most easy to understand. However, probably the least likely and the less efficient way is direct binding. So another way that it could happen is represented by this one. And this is really kind of called facilitated diffusion. So what this protein would do is it would bind into the major groove of DNA, and it could move in either direction. However, it would move in a 2D way. It wouldn't be out in space. It would be limited only to the stretch of DNA, and it would move along and diffuse to this 2D site. So no matter where it binds along the DNA, it can move in either direction, but it could still always reach this target sequence. So this is a lot more likely. However, it still has its issues. So there's still going to be other proteins that are bound to the DNA. DNA isn't naked, it isn't bare. So if this protein is trying to just 2D linearly diffuse across, it could interact with these other proteins and get knocked off. It could knock off other proteins. And also, how would it go around nucleosomes? Would it have to rotate around every time? So another issue is, what about how much of a difference is there between the binding interaction strength of the target sequence versus some other random sequence? There's a high likelihood of this target sequence being very similarly found in another section of the genome. So there's also problems with this one. However, it creates this, it decreases its diffusion by limiting it to 2D space rather than 3D space. So finally, we have this protein, and usually it was found in P53, and it's called kind of the DNA walk. And what it allows this protein to do, it can undergo facilitated diffusion. However, it can also jump. So it can move from one chromosome to another. It can move non-specifically. It can kind of scan proteins, which is kind of the facilitated diffusion section, or it can jump over nucleosomes, which kind of overcomes the battle of running into other proteins. It can jump over other proteins. And these proteins are usually have this core section, and it also will have these flexible loops coming off of it. And those flexible loops are usually the sections that are going to be interacting with the DNA. However, this also still has the same kind of problems. What kind of differences does it have in binding, interacting, the interaction binding strength of the DNA from the target sequence versus the other sequence? And how does it know when to jump between chromosomes? How does it know where to go, really? What is it directed by? And what I found between both the facilitated diffusion and the P53 DNA walk is that it depends both on the DNA size, about which one it will undergo, and it also depends on the target sequence size. So that's kind of the mechanisms that proteins would bind. And so usually, again, you would have, how we talked about, it would be sequence specific, and these would all bind in the major groove. Okay. Now we have base flipping, and these are specific proteins that will bind to DNA in the major groove as well. And what happens is you have a target base that you have, and it's going to be rotated completely out of the double helix. So you're going to be breaking those Watson-Crick base interactions and rotate it all the way out. So, and this is going to be done whenever the protein needs to undergo functions to perform in the DNA that can't be undergone in the helix. So usually you're going to be modifying the nucleotide or the base in some kind of a way, or maybe even the three ways that we've talked about is chemical modifications. So things like methyltransferases, phosphorylation. It could be for DNA repair. So in this case, this is T4-endo-V. What this does is it repairs thymine dimers. And you could also have ones that remove uracil, like uracil DNA glycosylases. And what these do is they're needed to undergo base flipping because you have to gain access to this really narrow space. And so the only way to do that is to flip out a whole other base. So in this case, with this methyltransferase, you're going to be flipping out the base that you're going to be acting upon. So here's the base right here. This is a part of the DNA, and it's going to be methylated by this S-adenosyl methionine. And so the flexible loops are the ones that are interacting with this protein, so you can methylate this one. In this case, the red section is going to be the thymine dimer, and you're going to flip out this adenosine so you can have better access to the thymine dimer. So in either way, you're opening up to gain access to this other space. Some other cases that you might also need to undergo base flipping is to nucleate strand separation. And so there's not necessarily crystal structures of these events happening, but these are still some probable hypotheses. So if for things like helicases and RNA polymerase sigma subunits, you could have these proteins that will actually flip out a base just to nucleate the strand separation to undergo its functions. So actually a fourth scenario that could undergo base flipping is to stabilize the protein DNA complex. So if you need a protein to bind really strongly and you want to kind of artificially increase the binding affinity, what you can do is actually flip out a base and stabilize that interaction, and that'll keep the protein landed on the DNA. It'll prevent its diffusion away. It'll stabilize the protein DNA interaction. And this could be done for things like transcription factors or proteins that will enhance chromatin structure. Okay, so this last section is something that is undergoing in prokaryotes, but I still thought it was interesting. It explains how DNA binding proteins can actually impact the genome structure in the order of genes. So in prokaryotes, we know that transcription and translation is coupled. And so knowing this, it actually creates this pressure for the co-localization of genes with a low copy number. And it shows that spatial organization impacts the levels of gene expression. So whenever you have this gene, specifically it's for transcription factors, but it could also apply for other DNA binding proteins. What we have is RNA polymerase. It could transcribe this gene. Once you have it translated, since these are done together, they are all done together, what you have is this protein will be still right there. So it limits the diffusion. This transcription factor doesn't have to diffuse away. Its binding site is directly there. It's right there. And so this can also be shown to have an impact possibly in signal transduction pathways in eukaryotes. However, it's only under the same principles, not exactly the same thing with understanding spatial organization and genome structure. And so by having this transcription factor close to the DNA site that it binds, it allows for these genes to not have to be copied as much, right? You don't need as many copies of the transcription factor if you're going to have a bunch of them successfully bind to the desired binding site. And so that makes things really efficient. You always want to be able to limit diffusion so you can make every process more efficient. So I think this is a cool example, although it only does apply, unfortunately, to prokaryotes since they have transcription and translation are coupled. So thank you.